Good morning, grade 12 students. In this session, we're going to continue with the exercises of chapter 6 about the roles and the components of the immune system. In exercise number 4, they said that in 1974, Zinker demonstrated that the double recognition of peptide HLA complexes by the TCR. In the TCR, they recognize it by a process called double recognition. They recognize self HLA, then they move to the antigen. هذه سميناها double recognition process. Based on the following experiment, mice of strain X were infected with a disease called CML virus. This one is choriomeningitic leukemia virus. This is the CML. This CML causes serious and not fatal cerebral disease. 1 week later, T cells from the infected mice were collected and put in culture. يعني أخذنا T cells from the infected mice. They are collected and they are put in culture, and in the presence of infected or non-infected mouse cells, either derived from the strain X or from a different strains. For example, as a Y strain. يعني أنا هون عم باخد strain Y. Strain Y who different from strain X. What does this mean? This means that they have different HLA. So they have different HLA molecules. The following results were obtained. First of all, we have infected X cells. حطينا infected X cells، حطينا معهم the T cells that are derived from infected mice لأن already عندهم هيدا CML. لما حطينا infected X cells, so these infected X cells يعني infected cells from the mouse of strain X حطيناهم مع T cells from the X mouse he already affected. So صار في عندي lysis. However, إذا حطينا non-infected cells اللي هن الX cells ما صار في عندي lysis. What does this mean? This means that the T cells, they cause the lysis of infected cells and not the non-infected cells. This is the first note you have to know. The X cells that are infected, they are lysed. However, the non-infected X cells, they are not lysed by the T cells. I did the first important note. Second important note, حطينا Y infected cells. يعني جبنا infected cells from another strain هو strain Y. حطيناهم مع TCR أو مع T cells from infected mice. ما صار في عندي lysis. Why? Because the T cells أنا جبتهم من strain X, strain Y هو different strain. يعني they didn't recognize the same HLA. يعني ما في عندي recognition of the HLA. That's why ما حيصير في عندي lysis. As well, why non-infected cells? So non-infected cells جبتهم من strain Y. طب non-infected cells من strain Y هيدا شو بيعني؟ هيدا بيعني انه non-infected strain from cells from strain Y. This means that these cells that are not infected كمان هن موجودين من different strain. So what does this mean? This means that these strains that are not infected ما عندهم infection وهن من different strain. So what does this mean? This means that there is no lysis لأنه ما في عندي HLA. So هون we can conclude that the recognition or the double recognition process of the T cells, it should be accomplished by the same HLAs. Again, under different HLA, this process won't occur. So conclusion for this, or the solution for this, when the medium contains the cells of immunized X with infected. What the meaning of immunized? Immunized meaning that they have X cells that are immunized, meaning that they have the T cells from infected mouse. يعني جابوهم من cells already infected كانت with the disease CML. That's the meaning of immunized. يعني عندنا المناعة against the CML. So there is cell lysis. So عندي lysis of the infected cells. However, if the medium contains T cells of immunized mouse X, T cells of immunized mouse X with non-infected X or Y. So عندي X or Y اللي هن non-infected. ما صار في عندي lysis. ما صار في عندي lysis for the cell. What does this indicate? This indicates that the T cells, it recognizes and it causes the lysis of the infected cells, but not the non-infected cells. However, if the medium contains T cells of immunized mouse X with infected cells of the strain Y, يعني هدول different strains, they have different MHC, different major histocompatibility complex. So there is no lysis. What does this indicate? This indicates that the T cells, they recognize and they cause the lysis of infected cells having identical MHC. يعني هنا بيعمل لايسيس للسيلز اللي عندهم نفس ال HLA او نفس ال MHC. So here conclusion, the antigen recognition by the T cells, it follows, it needs or it necessitates the presentation of the antigen of self MHC. لازم يكون عندي first self HLA or self MHC. إذا ما كان في عندي, this process won't occur. This what is known by the double recognition of the antigen and the self MHC. إذا حكينا عنها yesterday, it's called the double recognition process. The double recognition, it has to recognize the self HLA and then it recognizes the antigen. So this is called the double recognition process. That's why ما قدرت تتعرف على infected cell from the strain. Why? Because they have different HLA, and this it follows the double recognition process. Now moving to exercise 7. Exercise 7, we cross mice of two different genotypes. Okay? A strain of haplotype. They have haplotype مثل الجينوتايب بس is for a haploid cells. So strain genotypes, واحدة عندها HBB, second one عندها KK. A strain of haplotype KK والsecond one عندها BB. So بسموها H2B or H2K اللي عندها 2K أو عندها 2B. In the next document, MHC antigens that are expressed on the membrane of lymphocytes, they are represented by squares. يعني هون عندي أنا BB, هون عندي KK. So here صار عندي representation of B and K. هون بعطيكم note قايل لكم what is the haplotype is the set of genes on a chromosomic region carried by one of two chromosomes. يعني هي مثل الجينوتايب from haploid cell. So here بينوا اثنيناتهم. يعني هون أخذ from the maternal and from the paternal origin. So here expression of the MHC, it obeys the law of codominance where both of them, the maternal and the paternal, they are expressed. هيدي الأيديا اللي بدي إياك تعرفها هون. The expression of MHC, it obeys the law of codominance where both of them, they are expressed. هيدي اللي بدي منها هيدي الفكرة. So after crossing homozygous mouse having haplotype BB with another homozygous mouse having haplotype KK, شو طلع عندي؟ They gave a mouse having haplotype both B and K. So both haplotypes, the maternal from the mother and the paternal from the father, both of them, they are expressed. So here we can deduce that the expression of the MHC, it obeys the law of codominance. What does this mean? This means that both of them, they are expressed. Moving now to chapter 7, which is the last chapter. Chapter 7, activity 1, about the non-specific immune response. We started about this, and we talk about the non-specific immune response. Here about the non-specific immune response, and it doesn't depend on the identity of the intruder. It includes the phagocytosis process, the non-specific immune response. Before we start, let's make a quick revision about the immune response. Immune response, it's of two types. First type, it's a non-specific immune response. It does not depend on the identity of the intruder. Second, effector cells, they are monocytes and granulocytes. What happened during the non-specific immune response? We have the inflammation and phagocytosis. And you have to know that this non-specific immune response is local. It's a specific region in the body, for example, at the level of the skin, of my hand, for example, at the level of my face, for example. This is meaning of local, موضعي. However, the specific immune response is the second type, specific. It depends on the identity of the intruder. The effector cells in the specific immune response, they are the B cells and the T cells. While the specific immune response is systemic, meaning that it occurs all over the systems of the body. You have to know that the specific immune response composed of two different kinds. The first kind is called the humoral, and the second one is called the cell-mediated. We said that the humoral immune response is carried out by B lymphocytes, and it includes the secretion of antibodies where inside the serum. However, the cell-mediated immune response, it's carried out by the TC cells. TC cells, if you remember, T cytotoxic cells, where the TC cells, T cytotoxic cells, they kill the infected cells by the secretion of the perforin and granzymes. So you have to know this. Humoral and the cell-mediated, they belong to the specific immune response. Let's start with document 1, the non-specific immune response. When a pathogen crosses natural barriers inside the body or the tissues in the host, it triggers the host defense mechanism without few hours. The first phase is called the non-specific immune response. Before we start with the non-specific, let's remember together what we talked about in the introduction for the immune system. We talk about the natural barriers of the body. Natural barriers, they include what? They include the skin, they include sweating, they include, for example, the mucosal secretion, the saliva, the mucus, for example, the acidity in the gastric juice in the stomach, also the vaginal secretion in the female and the semen in the male. They contain antimicrobial substances. All of them, they are classified as the natural barriers. What about the manifestation of a non-specific immune response? Few hours after an injury, the infected one becomes red, swollen, and you are going to feel hot and painful. These are the signs of inflammation: redness, heat, edema, and pain. Edema, which is the swelling or the swelling of the infection. These signs appear in the case of many infections, such as, for example, the dermal otitis, for example, ear infection, pharyngitis at the level of the throat. The physiological events leading to these signs are broadly referred to as the inflammation. So when a microbe enters a tissue, infected cells like microbes and other white blood cells, they are present. They release a variety of chemical messengers. هدول منهم يا رايح نسميهم these messengers. They are called cytokines. What are the cytokines? You have to know this. Cytokines, they are small soluble molecules. They act on different target cells and they have local and systemic effects. What is the function of the cytokines? Cytokines, they cause larger enlargement, if you want, of the blood vessels. They increase in the diameter, leads in the increase in the blood flow to the site of inflammation. Also, cytokines, they have an important role. This here is the roles of the cytokines. These cytokines, they cause an increase in the vascular permeability and the leakage of plasma from blood vessels to the surrounding inflamed tissues. They move from one side to another. Other cytokines, they are chemotactic. What does this mean? They attract the leukocytes. What are these leukocytes? Mainly, they are the phagocytes. They are monocytes and the granulocytes, where they cross the capillary walls and they move toward the infected tissue. Hydrolysis process, this occurs mainly during the inflammation at the level of body. So this inflammation reaction and it's followed by phagocytosis, where it's a process of a non-specific immune response in the phago, meaning eating other cell. It's sufficient to eliminate the intruder. The phagocytosis process includes three important steps. The first step is the adhesion. Second is the absorption. Third is the digestion. These are the different steps and the different steps of the phagocytosis process. Three different steps. First of all, we have adhesion. Second, we have the absorption. And the third case, we have the third step, sorry, we have digestion. We have three cases. Cases are, we have, for example, we have deficiency in the enzymes, or maybe the bacterium is resistant to phagocytosis. So what will happen in these cases? Let's see. So here, this is the step of phagocytosis. Already we talked about them before. So first of all, I want you to solve the probing the documents on page 139. So what is the natural barrier? The natural barrier is an organ or a tissue, or it may be a secretion like the nasal secretions, or the mucus, or the vaginal secretions, or the semen. They prevent the entrance of microbes or antigens inside the body. Number three, the set describe the different steps of phagocytosis. Phagocytosis, as I said, they include three steps. We have adhesion. absorption and digestion. First, during adhesion, the macrophage approaches toward the microbe and adheres to it. The plasmatic membrane encloses the microbe. Next step عندي شيء اسمه absorption, where the absorption of the microbe occurs and the microbe is absorbed into the cytoplasm of the phagocyte and is enclosed in a vesicle containing lysozymes. Lysozymes, they cause the lysis of the cell. Finally, digestion of the microbe took place. Number 3, they are talking about, we have 3 different cases in the bacterium. First case, the bacterium is digested, bacteria waste are rejected, and the infection is going to be stopped. This is the first case. Whenever we inject, the bacteria is digested, the waste of the bacteria are rejected and released outside, and the infection is stopped. However, in the case there is enzymatic deficiency. What does this mean? This means that we have enzyme deficient. We have enzymes that are not present. So this bacterium is going to remain. Either is the bacterium is resistant also the bacterium will stay and it can multiply more and more. It's a stationary phase as in the second case, or the bacteria multiply, the nucleus of the phagocyte will disintegrate, lipids and granules will appear, and this will lead to the appearance of the pus. يعني العمل اللي بيطلع وقت الجرح أو وقت الالتهابات and so on. So pus is observed and the infection is continued here whenever we have enzymatic deficiency or we have a resistant bacterium. Phagocytosis, they said it's non-specific, number 4. They said why is phagocytosis referred as non-specific and formulate 2 hypotheses explaining why some foreign bodies may resist phagocytosis during the non-specific immune response. First, why phagocytosis is non-specific? Because it does not depend on the identity of the intruder. Why some bacteria they are resistant and they persist during the phagocytosis? Maybe we have enzymatic deficiency, that's why the bacterium is not digested, or this bacterium is resistant. So mainly these are the proposed hypotheses to explain why the bacterium they are resistant during the process of phagocytosis. Now, document 2, we're going to talk about the specific immune response. Specific immune response. We said that when the non-specific immune response fails, we have to introduce the specific. This response is adaptive, meaning that the components, they can identify any intruder and they can respond to it. Here, pay attention, يعني هون بلش الجد في الدرس. Experimental study. Vibrio cholera and Salmonella, they are 2 types of bacteria. They can cause serious intestinal infection. Experimental injections of live Vibrio or Salmonella to animals are fatal. What the meaning of fatal? شو يعني fatal? What the meaning of fatal? Yes.
Fatal, it means dangerous.
Fatal, not dangerous Muhammad. Fatal meaning that it can cause death. يعني بتقتل.
Deadly.
Okay? Deadly. يعني it causes death. So when the meaning of fatal, it's that it causes death. Okay? So عم بقول لي injections of live Vibrio cholera or Salmonella. يعني إذا أنا حطيت لك هلا أنا جبت لك Salmonella عملت لها injection, it may cause death. إذا كانت عايشة. إذا كانت killed, it's like the case of vaccine. يعني أنا عم بعطيك immunity against the Salmonella or against the Vibrio cholera. So here they did an experiment. If they are submitted to high temperature, the bacteria die because their antigens are preserved. Let's start step by step in the experiment. هبلش step by step in the experiment number 1. In experiment number 1, what did they do? Experiment number 1, كان عندي جايبين an animal, for example, whom they injected with killed Salmonella. So عطوه killed Salmonella. Killed Salmonella يعني هلا انتم عم تسمعوا في الكورونا فيروس فاكسين. هلا mainly الفاكسين على الكورونا فيروس somehow different. So الكوفيد 19 vaccine somehow عم يشتغلوا فيهم بطريقه ثانيه، عم يشتغلوا at the level of adenovirus، عم بحطوا spike proteins and others. So عم بحكي about for example flu virus. Flu virus, الفلو فاكسين له بيعطوك الفلو virus but it's a very very low amount او بيعطوك اياه killed flu virus. يعني انت you're going to recognize the antigen, your body will recognize it, will form memory cells against it, and your body will resist and will attack it. So جسمي حيتذكره بس يفوت مره ثانيه مش معناتها ما بقرب بس بيكون قريب كثير خفيف لانه already I realize this antigen as a non-self and you kill it. So here I killed salmonella. فوت السالمونيلا. So my body recognize this as a non-self. So لما تفوت ترجع السالمونيلا after 10 days فاتت حط لي لايف سالمونيلا، يعني سالمونيلا هون عايشه، يعني هون it can cause death، فيها تقتلني، بس انا جسمي has the immunity against the salmonella since killed salmonella was injected. So killed salmonella provide the body with immunity against the live salmonella and this will lead to the survival. However, second experiment استعملنا Vibrio cholera، عملنا injection of killed Vibrio cholera، اعطيتها للانيمال killed Vibrio cholera. Then 10 days اعطيتها live Vibrio cholera، Vibrio cholera العايشه. شو صار فيها؟ the animal survived، ده العايش. So هون kill Vibrio cholera حمتني من اللايف Vibrio cholera، والكيل salmonella حمتني من اللايف salmonella. Now let's move to experiment 3. Experiment 3 I injected killed salmonella. So هيدي الكيلد salmonella حتحميني من اللايف salmonella. 10 days عملت injection of live Vibrio cholera. حطيتها شيء ثاني، حطيتها Vibrio cholera عايشه. شو صار فيها للانيمال؟ ماتت. However in experiment 4 حطيتها killed Vibrio cholera بس جات after 10 days حطيتها live salmonella. So ماتت. Why? Because killed salmonella ضد مين بتحميني؟ against the live salmonella while the killed Vibrio cholera تحميني against the live Vibrio cholera مش against the live salmonella. So هم بتفرجيني that the immune response is specific. يعني الانتي بودي against salmonella بيحميني ضد السالمونيلا ما بيحميني against Vibrio cholera. نفس الشيء اذا اخذت انا فاكسين ضد الكورونا فيروس ما بيحميني من الايدز for example. So this shows that the immune response is specific. So هون عم يحكي عنا how do the experimental results of document A show that the immune response developed 10 days after injection of salmonella or Vibrio cholera is specific. Part number 1. أنا عملت interpret. Chicken that is injected with live salmonella 10 days after being injected with the killed salmonella survive. At the same time, chicken that injected with live Vibrio cholera 10 days after being injected with the killed salmonella it dies. So what does this indicate? This indicates that the killed salmonella تعطيني specific immunity against salmonella اللي هي live salmonella, not against the live Vibrio cholera. As well as the chicken that is injected with live Vibrio cholera 10 days after being injected with killed Vibrio cholera, it survived. The animal survived. At the same time, chicken that is injected with live salmonella 10 days after being injected with killed Vibrio cholera, it dies. What does this indicate? This indicates that killed Vibrio cholera provides specific immunity against Vibrio cholera and not against salmonella. يعني الانجكشن of killed salmonella protects against the salmonella and the injection of killed Vibrio cholera protects against Vibrio cholera. Types of specific immune response. Okay? The specific immune response develops usually within 1 to 2 weeks following the contact with the foreign body. This, in order to understand what type of specific immune response is induced against a given antigen, the following immunity transfer experiments are performed. These experiments consist of inducing a specific immune response against a microbe or a toxin. What the meaning of toxin? Toxin that can cause diseases and maybe it's toxic. Toxoid, pay attention to this word. Whenever I said toxoid, يعني هو المخفف. يعني أنا for example, tetanus toxin. Okay, pay attention to this note. If I said, for example, tetanus toxin. Tetanus toxin, tetanus toxin. So tetanus toxin is toxic. While if I said tetanus toxoid. توكسايد معناتها مخفف كتير، يعني كأنه عم بعطيها immunity، يعني كأنه عم بعطيها فاكسين، يعني هي provide immunity. Also كمان إذا قلت attenuated، attenuated كمان معناتها مخفف كتير، يعني it's very very weak. This is the word attenuated. So keep this in your mind. Attenuated or toxoid, for example, is amara omana. Attenuated or toxoid meaning that it's very 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 weak. يعني it can cause that. يعني ما بتقدر تقتلنا even though maybe sometimes it can cause a disease. Okay? So here the recipient subsequently receive an injection of that microbes or toxin that's normally fatal. According to the outcome, one can determine which blood component provide protection. نحن حكينا إذا السيروم عمل لي protection يعني أي type of lymphocytes. Let's see لنشوف إذا متذكرين أو لا. إذا أنا حكيت لعلي حريري قلت serum provide the protection sorry. So which type of lymphocyte is working here? إذا حكيت serum. Yes. What do you think, يا علي حريري? Is the added serum provide the protection against a specific toxic. So what does this mean? This means that which type of lymphocyte, B or T? إذا حكيت serum. Great. Hassan Watfa, what do you think?
B lymphocyte.
B lymphocytes. Why? Why it's B lymphocyte? هذا السؤال صار صعب هلا إيه؟ Because in the serum we have antibodies, and antibodies they are secreted by B lymphocytes. That's why they are the one that protects. Okay, so let's move. In experiment A and document B, what did they do? We study the immune response against tetanus toxin. Subhanallah, بعدنا عم نحكي عنه. Tetanus toxin that is secreted by the bacteria Clostridium tetani. This toxin is responsible for tetanus. This can be fatal. Laboratory manipulations eliminate the harmful impact of the toxin without altering the antigenic determinant. مين في يقول لي شو يعني antigenic determinant؟ امبارح بعد ما حكينا عنه. What does the antigenic determinant mean؟ شو يعني antigenic determinant؟ شو هي الكلمة غير antigenic determinant؟ Great, great. قلت له بدك تعمل quiz this week, yeah? So great. This antigenic determinant بنسميها yes Selena.
epitope.
Epitope, very good. Thank you Selena, very good. So antigenic determinant هي الابيتوب. يعني أنا هون شو عملوا؟ عملوا laboratory manipulation eliminated the harmful effect of the toxin. يعني عملوه كثير مخفف بس خلوا الابيتوب تبعوله. يعني أنا إذا تعرف عليه الأنتي بودي يتعرف على الابيتوب. يعني إذا ضلت الابيتوب كونستنت ما تغيرت، ما غيرت الابيتوب، إذا فات مرة ثانية وكان هو الهارمفul فيي اقتله دغري لأنه أنا identified the epitope, the part of the gene. هي بنسميه antigenic determinant. So هون the neutralized toxin يعني المخفف، التوكسين المخفف او اللي سميناه توكسويد او اللي سميناه attenuated هون هو بيكون كثير خفيف. So first of all شو عملوا باكسبيرمنت A؟ They injected attenuated toxoid. هيدا attenuated toxoid يعني هو tetanus toxoid هو اللي مخفف. And then what did they do? They took the serum and they injected into mouse A2, and they took the blood cells and they injected into mouse A3. هيدا اعطوها السيرم وهيدا اعطوها blood cells. Okay? لو اعطوها السيرم it survived after the injection of tetanus toxin اللي هو harmful. بينما لو اعطوها blood cells it died after the injection of tetanus toxin. يعني مين اللي حماها؟ حماها السيرم. يعني اللي حماها السيرم this means that the lymphocytes اللي حموها. Okay? هون tetanus toxide it was attached to a solid matrix and then they injected tetanus toxin. So هون علقوه بsolid matrix and then they injected with tetanus toxin, it died. However, in another experiment, in experiment B, في عندي disease اسمه CGB. CGB هو a disease that is caused by the harms. It becomes CGB هو the harmless one. So CGB هو في عندي بكتيريا اسمه كوك باسيلس. كوك باسيلس كي بي هي responsible for tuberculosis. Tuberculosis هو infects the lungs وبيعطي problems على lungs and they lead to death. كالمن اند جيرلين باسيلس سي جي بي هو هارمس بكتيريا اللي عنده نفس الكي بي، نفس سوري الابيتوب مع الكي بي. يعني الكوك باسيلس والسي جي بي عنده نفس الانتيجينيك ديترميننت، بس هيدا السي جي بي هو not harmful. يعني اذا انا اعطيته السي جي بي ورجعت اعطيته الكوك باسيلس الكي بي ما حيموت لانه بيعمل له بروتكشن. سو انا اعطيته سي جي بي، اوكي، سي جي بي، سو عنده protection against the cock bacillus. then انا اخذت السيرم وعملت له injection with another mouse B2، اعطيته كي بي لهيدا الماوس on meds. however اخذت البلاد سيلز اعطيتهم لهيدا الماوس B3 واعطيتها كي بي it survives. فمين اللي حماها؟ حماها الليمفوسايتس. رجعت انا عملت separation for the lymphocytes، اخذت البي سيلز لحال والتي سيلز لحال. لما اعطيته بي سيلز ورجعت اعطيته كي بي died. لما اعطيته تي سيلز واعطيته كي بي survive. مين اللي حماه؟ حماه هو التي lymphocytes مش البي lymphocytes. فهون انا حل part 2. I describe the experiment. هعمل لها describe part 2 what can it reduce and what is the type of the immunity present. So description هو مش طالب describe بس انا عم ذكركم فيهم. At first, mouth A is injected with tetanus toxide, then its blood is collected and separated into serum and blood cells. The serum is separated into two parts. The first part is injected into mouse A2 with tetanus toxin. It remains alive. Then the serum, the second part, is filtrated into a tube containing tetanus toxide, attached by a solid matrix, and then injected to a mouse with tetanus toxin is done. It dies. Then the cells are injected into mouse A3, the blood cells, it dies. Interpret. Mouse A2 that received the serum of immunized mouse. يعني immunized يعني أخذت tetanus toxide with injected with tetanus toxin it survived. At the same time, A3 that received the cells of immunized mouse A1, it dies. This indicates that the effectors against the tetanus toxin موجودين inside the serum and that the cells. However, mouse A4 that received the serum of mouse A1 after being filtrated into a tube contained tetanus toxide and then injected by tetanus toxin, it dies, meaning that the effectors against tetanus substances that are found inside the serum and they are bound to tetanus toxin. That's why راحوا هدول substances and that's why it dies. However, in document B, mouse B1 is injected with the CGP and the CGP هي harmful bacterium but عندها نفس الابيتوب مع الكي بي مع الكاك باسيلس اللي هي harmful and causes tuberculosis or it causes death. Then the blood are separated into serum and cells. The serum is injected with KB into mouse B3, it dies. The cells separated into two parts. First part we injected into mouse B2 with KB, it survived. The other part is divided into two parts, B cells and T cells. B cells injected B5 into with mouse B5, it survived. So on mouse B2 that received the cell of immunized B1 are injected with KB, it survives. At the same time that received the serum, it dies. This indicates that effector cells against KB, against the bacillus, they are found inside the cells and not the serum. And then we have to indicate that the effector cells against KB in the T cells ومش ال B cells. آخر شيء عملت conclusion، يعني عملت هيدا البارت أنا describe وعملت interpret وعملت له conclusion. So against tetanus toxin, the immune response is humoral, since the serum is the active, while against the cat bacillus, it's the humoral, the cell-mediated immune response, and not the humoral, because the cells they are involved, and mainly the T cells they are involved. Okay, so here we finished document 1 and document 2. And the next session we're going to do about document 3, induction of the specific immune response, and we're going to talk about document 4, the role of T helper cell in the specific immune response, and we're going to move to document 5, the specific humoral immune response and specific cell mediated. Already we covered all of them. And document 7, we're going to talk about the immunological memory, and document 8, we're going to talk about diagnostic applications of the antibody property. This is the last chapter and the last documents. Then we'll move to the exercises. So next week mainly will be the last week in this chapter.